If you've ever wondered about the molecular basis for recombination between DNA, here are the three models. The first model of recombination was the holliday junction double-strand invasion model. Holliday is the man who created it. A holliday junction is a formation where the two DNA molecules are ligated together but have not yet recombined. Double-strand invasion means that both strands invade or enter and bind to each other. Here is the model. First, the two double-stranded DNA molecules each get a single-strand nick on opposite strands. These new ends then ligate with each other, creating an X. The strands get uncrossed and bend out to form a holliday junction. The holliday junction can then isomerize to form two different configurations, 1 and 3, which then resolve or break apart into two double-stranded DNA molecules to make different recombinants. The problem with this model is that recombination can also occur with one single-stranded DNA molecule, so this gave birth to the single-stranded invasion model. First, the double-stranded DNA molecule gets nicked. The exposed end of the DNA at the nick breaks off a bit and runs through another double-stranded DNA molecule, breaking its bonds, or across a single-stranded DNA molecule until it finds a complementary sequence. The exposed end binds to the complementary sequence, and the other strand of the second double-stranded DNA molecule is degraded. DNAP uses the exposed ends of the nicked DNA to resynthesize DNA using the remaining strand from the second molecule as a template strand. This is the double-strand break model. First, one double-stranded DNA molecule gets a double-strand nick. The 5 ends of the nick are degraded down so that it appears like a staggered nick in the DNA. One 3 end of the nicked DNA invades the second DNA molecule, displacing but not degrading one strand as it searches for a complementary sequence. Once it finds a complementary sequence, it uses the displaced strand as a template strand to resynthesize DNA on the top and the sides of this new molecular structure, thus forming the junction.